we're going to do now is look at an overview, just sort of a general look at adaptive immune responses. Okay. Now, as we talk about adaptive immune responses, we do have to mention the topic of self-recognition and how our immune system distinguishes self versus non-self. Okay. So, distinguishing self versus non-self is something that the innate immune system did accomplish. Okay, so we've been talking about innate immunity, but when we talk about the adaptive immune responses, it's even more important to talk about self-recognition. So the most important self-antigen that we have is a set of proteins that are referred to as the major histocompatibility complex proteins. Okay. Now, there's a couple of terms that are used interchangeably. Major histocompatibility complex, or MHC, is often used to refer to the self-antigen. However, there's another term called the human leukocytic antigen, or HLA, human leukocytic antigen. Now, technically, the major histocompatibility complex actually refers to the set of genes on our DNA that codes for the human leukocytic antigen. Okay? So technically, the MHC, or the major histocompatibility complex, actually refers to the genes, and the HLA, or human leukocytic antigen, actually refers to the self-antigen. Okay? However, it's very common, and it's more common to hear the proteins that are our self-antigens as the MHCs, described as the MHCs. So that's what I'm going to do. So there are two types of self-antigens. We have MHC1s and MHC2s. And these MHC1s and MHC2s are expressed by different cells, and they signal different things. So first off, the MHC1 proteins. Now, MHC1 is the self-antigen that we find on all cells in our body except for red blood cells. Okay? So all cells in our body except for red blood cells have MHC1s. And the MHC proteins, so the genes that are coding for these proteins, are the most varied sets of genes among individual human beings. So from one person to the next, we are not all that different in terms of our genetic makeup, but the major histocompatibility genes are the most diverse among human beings because they have to indicate one person from the next. Okay? So one person's MHC proteins are not the same as the next person's MHC proteins. They are different enough to be unique. So we have MHC1 proteins on all cells in our body except red blood cells. Okay. Now, what MHC1s, they start off as intracellular proteins. So the cell produces this MHC1 protein, and then before the MHC1 travels to the membrane, it binds to other proteins being produced by the cell. Okay. And when it gets to the membrane, it displays that protein on its surface. And so the purpose of that is this cell of our body, okay, this cell of our body is signaling, I belong to you, okay, I have this MHC1, so I am a cell that belongs to you, and this is the type of protein that I am making right now. Okay? So that's what that says. It says, I'm a cell that belongs in your body, and this is the type of protein that I'm making right now. And this signal is designed to be read by two types of cells: the natural killer cells and the cytotoxic, or CD8 T cells, because these are the cells of our immune system that are responsible for recognizing abnormal host cells. And by abnormal, I mean virus-infected or potentially cancerous cells. So if the host cell is, let's say, virus infected, then the MHC1, before it gets to the membrane, is going to bind to viral protein. And it's going to wave that viral protein in the air, and the cytotoxic T, or the natural killer, is going to recognize this is a host cell that belongs in my body, and it is making viral protein. So now it's going to then kill that cell. Okay? So that is the MHC1. It's present on all cells, and it's actually designed to communicate with the natural killer cells and the cytotoxic T cells. And it's designed to display self-recognition, I belong to you, and also the possibility of abnormal protein, which could signal an abnormal host cell. Now the MHC2s. The MHC2 proteins are not found on all cells in our body. In fact, they are only found on 3 very key cells in our body called the macrophages, the dendritic cells, and the B lymphocytes. And the reason why they are only on these 3 cells is because these are the 3 cells that carry out a very important job. This job is what we call antigen presenting, or antigen presentation. And antigen presentation is the key first step in initiating adaptive immune responses. Okay? So these are the 3 cells that can trigger adaptive immune responses through antigen presentation. Okay? So these 3 cells, these 3 groups of cells, contain both MHC1s and MHC2s. Okay? And they present their MHC2s for the CD4 or the helper cell to recognize them. Okay. So let's say there's a macrophage. Let's say a macrophage phagocytizes a pathogen. It's going to digest that pathogen. It's going to take the antigen on that pathogen, and it's going to bind it to its MHC2. And then that MHC2 carrying that pathogen antigen is going to travel to the surface of the cell, and it's going to find a CD4 T cell to present that antigen to. And when the CD4 T cell sees this macrophage, it recognizes the MHC2 as being self. It recognizes the viral antigen, or the pathogenic antigen, and it then initiates an appropriate adaptive immune response. Okay? So the MHC2 is a secondary MHC protein only found on these 3 types of antigen-presenting cells, and the MHC2 is key to communicating with the CD4 T cell and triggering adaptive immune responses. So what happens when we trigger an adaptive immune response? So we're just going to look at this from a very simplistic view, because we're going to look at the details of this later, actually in the next lecture. So this is a very simplistic overview of what happens when we trigger an adaptive immune response. So this is just a very basic description of the steps. So what we're looking at here, this yellow circle represents the antigen presenting cell, or the APC. Okay? And the APC, this could be a macrophage, this could be a dendritic cell, this could be a B lymphocyte. The antigen presenting cell is presenting antigen, okay? And the antigen in this slide is represented by this red triangle. It's presenting the antigen attached to its MHC2, and it's presenting it to the CD4 helper T cell. And it's presenting it to not just any CD4 cell, but the very specific CD4 cell that carries the T cell receptor that is complementary to this antigen. Right? So it is the red receptor that can bind to the red triangle. When it does this, the CD4 T cell recognizes the antigen, and it is either going to trigger a B cell response or a T cell response. If it triggers a B cell response, the first step is the CD4 T cell has to find the specific B lymphocyte that is complementary to this antigen. Okay? Meaning the B lymphocyte that has the B cell receptor complementary to the antigen. Okay? And so once it finds that B cell receptor, it's going to trigger that B cell to make many, many copies of itself. One set of copies is actually banked as memory cells. So one set of copies of this B lymphocyte is actually going to just sit in the lymphatic system for the next time we encounter this pathogen. Another set of copies of this B cell are going to be what we call the effector set, meaning these are the cells that are going to fight the current infection. These B lymphocytes are going to undergo their final differentiation step to form what are called plasma cells. Plasma cells are the terminally differentiated form of the B lymphocyte, and they are the ones that actually secrete the antibodies needed to fight this pathogen. Okay? And so those antibodies enter circulation and they start going off and trying to find the pathogen. Now, let's rewind and go back to this CD4 T cell. When it is presented with the antigen, if it decides that we need a T cell response, then this CD4 T cell is going to find the appropriate CD8 T cell. Okay? And it's going to be the CD8 T cell that is specifically complementary to this antigen. When it finds that T cell, it's going to trigger that T cell to make many copies of itself. Okay? Again, one set of copies are going to be banked as memory cells for the next time we encounter the pathogen. And another set of copies are going to be the effector set, meaning the set of T cells that are going to then go off and try to find the pathogen and attack it. Okay. So these are the very, very general steps of initiating and mobilizing an adaptive immune response. So in order to get to that point where we can trigger an adaptive immune response, we actually have to back up a little bit and understand how we get the diversity of our B and T cells and what happens once we have that diversity, okay, and some key features of that. So there are a couple of key steps in the overall development of the ability to mobilize an adaptive immune response. And the two major steps are what's called the generation of clonal diversity and then clonal selection. Okay. So the generation of clonal diversity refers to the process of developing our huge library of B and T cells that can respond to any antigen that we encounter. Okay. So the generation of clonal diversity refers to that initial production of all of our B and T cells that can respond to every possible antigen that we will ever encounter. Okay. This process of the generation of clonal diversity happens in early development, okay, and to a certain extent throughout our lives, but mostly in early development. Okay. And the key aspect of this generation of clonal diversity is that we are going to produce all of these B and T cells before we ever encounter the pathogen. Okay? So we're going to make a T cell for a pathogen that we have never even encountered yet. Okay? Now, once we have our library of B and T cells, we then, when we do encounter a pathogen, we undergo what's called clonal selection, meaning we are going to select the clone of B and T cells that complements the antigen, that reacts to the antigen, and that's going to be the one that's going to fight the infection. Okay, so let's just talk a little bit about the generation of clonal diversity. So this is relevant for the production of our huge library of T lymphocytes and B lymphocytes. So the T cells, let's start first with the T cells. So we know that our T lymphocytes are first produced in the bone marrow, and they leave the bone marrow as these pre-T lymphocytes. And they migrate to the thymus gland, which is what is drawn here in this slide. They migrate to the thymus gland, and then they start to acquire all of the surface markers that are going to determine their functions. They're going to acquire either a CD4 surface marker or a CD8 surface marker, and that's going to determine their function. And they're also going to acquire a T cell receptor. Okay? And the T cell receptor is going to indicate the cell's specificity. That's going to be going to determine what antigen this T cell can fight. Now, the process of producing these T cell receptors, the reason why we can produce T cell receptors against antigens that we have never encountered is because the process is actually involves a lot of random reorganization of the genes that can code for the T cell receptor. So if we look at the genes that code for the T cell receptors, there are three basic sets of genes that we call them the V genes, the J genes, and the C gene. Okay? And so these genes are randomly rearranged, portions of the genes are randomly rearranged to produce a wide variety of specific proteins, of specific T cell receptors. Okay? And so that is how we can create such huge diversity in these T cell receptors that can bind to any antigen that we could possibly encounter. Now, because this is a total random process, we do run the risk of producing a T cell receptor that binds to a self-antigen. So we do run the risk of producing a T cell receptor that is what we call autoreactive, meaning that it can bind to our cells and try to kill our cells. Now, as these cells are matured, there is a process to eliminate those autoreactive cells, and this is called clonal deletion. So within the thymus gland, right next to these maturing T cells, we have epithelial cells throughout the thymus gland that display the variety of self-antigens that exist throughout the body. And these self-antigens are there so that if a newly formed T cell with its newly formed T cell receptor happens to bind to that self-antigen, okay, that is a signal for the thymus gland to destroy that T cell. Okay. So any T cell that binds to self-antigens of these epithelial cells in the thymus gland, they get deleted and destroyed. Okay? And that process is called, this process of clonal deletion, it produces what we call central tolerance, meaning that we tolerate ourselves. We do not produce T or B cells that are capable of fighting our own self-antigens. Okay? And the reason why it's called central tolerance is because it happens within these central lymphoid structures where these T and B cells are being produced and where they mature. Okay? So they don't leave the thymus gland carrying a T cell receptor that binds to a self-antigen. And we see a similar process in the production of B lymphocytes. Okay, we see almost an identical process. So the B cell receptor of B lymphocytes is coded by, again, a set of genes, V, D, J genes, that are randomly rearranged to produce the wide variety of B cell receptors, those IgM and IgDs, okay, that bind to any antigen in nature. And in the bone marrow, so this whole process is happening in the bone marrow, again we have epithelial cells that display self-antigen. So if any of these newly formed B cells binds to the self-antigens, they are deleted. Okay? So that produces the central tolerance involving B cell production and T cell production. So the B and T cells that leave the bone marrow and thymus gland, the mature ones, they carry B and T cell receptors that can, they're all naive cells. They have yet to encounter the pathogen they're destined to fight, but they are capable of fighting it. They are mature, and hopefully they are not autoreactive. They are not complementary to any self-antigen.